Live? Hey now. Cool. Perfect. I am going to move over to the document camera. We can draw a couple of pictures. Hi, everybody.
Hello.
It's great to see you again. It's great to see everybody again. Good fun. Hope you had a good. What'd you do yesterday, Giselle? You asked me what I did. I didn't know. What did you do yesterday? See, you don't know any more than I do. It's like,
Class.
yeah, which one do you remember?
Yeah, I went to my chemistry lecture and then lab, and then another class. History.
I'm in
And
the
then I
net.
stayed on campus until like 9. because-
Oh wow,
PM?
Yes.
Oh.
Yeah.
goodness,
It's
goodness.
like our finals kind of coming up, and like I'm
Yeah.
behind in some classes.
Yeah.
So,
Yeah. Are you behind you, or are you just exactly where you are?
no, I'm behind some chapters.
Well, there it is.
I suppose the question is behind what?
She's behind. Yeah, she's behind. It's like, no, literally, it's like 5 cents that need to turn in that are late. Maybe I am behind. I'm going to actually go like this. I'm going to flip this over. I'm going to draw the same picture twice. And the first time I draw the picture, it's going to be a linear version of the life of the cell. And then the second time I draw it, it is going to be a circular version of the life of the cell. Does that sound good?
Yes.
So it's like appreciating that both drawings are kind of the same, but different in that when we think about cells, in your textbook we'll talk about the cell, a lot of times they use this word, the cell cycle, as if it is some cyclical thing like the Krebs cycle or the Calvin-Benson cycle or any of these other cycles we sort of talk about. And then like it goes around and around in circles. To be clear, it does not. Time seems to move in a linear forward direction, right? So it's like the cell doesn't go like forward and then like back in time and goes, it really sort of doesn't work that way. So the conversation is oftentimes couched, even in textbooks, like the cell cycle, cyclical sort of thing, in that just like with the Krebs cycle and Calvin-Benson cycle, you get to the end and then you start the loop over again. It's like, great, that's what cells do. Also, they don't quite do it like that because time is, for our earthbound, gravitationally constant, mass-specific physics in which we live in, it's kind of uniformly forward, right? So it looks like, it's like, for something being a cycle, it sure looks like a line.
It's like a process.
Right, so yeah, so it's a process where you can tie the end of it to the beginning of the next go around, but that's not what happens here. None of my cells stopped and said, you know what, I'm gonna start over again, and sort of went back. So it's like, it exists, it's like light, it exists as photons, electrons existing as both particles and waves. Cellular lifespans exist as both lines and circles. So and I've always kind of brought curiosity, I almost said struggled, but brought curiosity. It's like, what's the best way to do this in a lecture then? And what I decided to do is, well, let's do it twice. One is a line, one is a circle. How do you describe an electron? As a line, as a circle? Well, I'll do it both. I'll describe it as a particle and I'll describe it as a wave. So this is how this might look. When we adjust my camera a little bit, I'm still surprised at how many people actually watch these videos. They rack up more than I thought they would.
Famous.
Yeah, well,
You're a content creator.
they're checking my LinkedIn, right, and making contact with me and all my cool stuff. I'm starting at the bottom and I work my way up. Is that okay?
Fine.
Perfect. I'm only doing it that way because that's the way I did it last year and it seemed to work. A cell on your body that's just kind of hanging out, doing its thing, living its life, being a part of your multicellular existence, is a cell that is in a phase of the cell cycle called interphase. I'll write that down. Interphase, these are your cells, most of them in your body, hanging out, doing like liver things and doing like kidney stuff and doing like pancreas stuff and doing like lung things. You know, most of your cells, most of the time, are hanging out in this. This is where life happens, in interphase. Cell goes on, cell lives its life, cell is doing things. Every once in a while, when some of your cells, a stimulus comes along and it says, cell, you know what you ought to do? Let's divide. And the one cell then divides into two. That process is called mitosis. Mitosis. And because interphase ends with phase, we're going to say that is called the mitotic
You know,
phase.
I literally don't say anything.
Mitotic phase. This, cells living their life, doing their thing. From here on up to the top of the page, cells going through the process of cellular division. Okay, the process of one cell becoming two. When a cell is doing this mitotic stuff, it is not doing the things for you. It's doing things for itself. All right? When cells are doing things down here, it is not doing this cellular division kind of thing. So cells really sort of, they exist either here or here. They're never sort of stuck halfway, with a couple of interesting, curious, almost exceptions, which are the subject for next week. Okay, but for the most part, cells are here or they are up here. And it's pretty easily to distinguish which one is which. Within interphase, down here, cell living its life, doing its thing, everything's great. I'm going to say this goes from there to there. You have this first, there's three components here. So just if you want to prepare your picture, I'll do some dots here. First one, second one, third one. The first component of this is called G1 or growth 1. That is a cell in its existence. It is doing life. So when I say most of your cells are in interphase, most of your cells are interphase, and they are in this very first part of this G1 part. Cells are just banging away, using ATP. Mitochondria is mitochondriaing. Glycolysis is glycolysising. Photosynthesis is photosynthesizing. It's being the cell that it is. Next one is referred to as the S phase. S phase stands for synthesis. Something is being synthesized in the S phase. And what that thing that is being synthesized is a lot of DNA. The DNA of the cell is going from being unduplicated to duplicated. That will be a separate picture that I will draw here in a moment. So I'm going to draw the whole framework and then I'll go back and fill in some deets. I doubted the C on that. Apologies. You double the amount of DNA in the cell when you do this. Okay, you're making copies. You're making some copies. G2 is next. Cell prepares for division. And I'll talk about what this is here in a little bit. I might talk a little bit about it now. cell makes everything it needs in order to divide. This is sort of the doomsday prepper version of the cell cycle, where it's like you start like hoarding groceries from Costco and things like that. And this is why. These cells are going to be alive and metabolizing and doing stuff all throughout the entire part of this. Once the cell starts mitotic phase, like the very first part of the mitotic phase up here, is all the DNA and all of the in the nucleus of the cell, all getting packaged up, wrapped up, condensed, kind of organized into tight little compact packages that are easily sorted out. When we're down here, all of your DNA is just unraveled, unspooled, 46 chromosomes worth of DNA, add them all together, it's about 6 feet long of strand-like material, all just sort of throughout the nucleus spooled out. If we divided those 46 strands, and I'm going to say, here, I'll grab 23, you grab 23, and let's pull, I'm just going to break them all, right? So in order to actually divvy these things up, you need to package them up in nice little sortable things that you can sort of move around in a neat way. The good thing about that is that it works. The downside is that once you start packaging up your DNA like that and bundling it up into little discrete packages like that, you can no longer actually access it to make messenger RNA, to make proteins with. So the DNA as a protein producing engine, a source of information, will shut down when you enter mitosis. So everything you need in that cell to get all the way through mitosis, to get back to the next interphase down the line, you need to have constructed to completion before you even begin this. Does that make sense? So it's like, make sure everything is in the car before you leave.
Can you go from the mitotic phase to the interphase, like when they're divided?
After they divide after mitotic,
Yes.
you'll have then two cells, which are interphase. That's how the line becomes a circle, right? Stay tuned.
No, but it's
You're about
a line.
10, 12 minutes ahead of me. Stay tuned.
So for, if everything is is it just DNA that's getting duplicated in?
It's not. It's other stuff too. If you think about one cell becomes two, both of those cells that you get out of it need to have the full complement of everything it needs in order to function. Right? So you have to duplicate payroll, you have to duplicate accounts receivable, you have to duplicate the mailroom, you have to duplicate,
Don't forget
admin, don't forget
legal.
legal, you have to duplicate marketing and advertising, right? You have to duplicate all divisions within the organization. So you have to make sure there's enough Golgi, you have to make sure there's enough smooth ER, rough ER, you have to make sure there's enough mitochondria to go around. You have to duplicate everything. So it's like, that's what happens here in this G2. It's like, I'm gonna make sure that I have enough of everything. You don't need to have it all made, but you do need enough to get started.
Right. So like, so S phase is specifically just highlighting DNA
The
duplication.
DNA duplication.
Okay.
And G2 is like, and all the other stuff that you might need to.
Okay.
Because if you think about it, when you were all but a fertilized egg in your poor mother's fallopian tube, remember that?
Yeah.
As if it was
Right.
yesterday. It was 51 years and 9 months ago, right? I was a young lad in my mother's fallopian tube even before I embedded into a uterine wall. I started dividing. And for those first, I almost said like, ladies, you know this, right? So for the first day or two or 3 or 4 ish, after fertilization doesn't happen in the uterus, it happens usually in the fallopian tube, right? And then a couple of days later, the embryo, right at that point, the cell, the plural, that are been fertilized are now embedding into the uterine wall, where it's going to form a placenta, and then humans going to grow inside of it. 9 months later, shabam, right? It's good. If you're a maternity ward doctor, every time you know, shabam, right? That's how you know it's over and done. Air horn, shabam, pop the thing. Shabam. So when that cell is just like working through the fallopian tube and entering the uterus for eventual implantation of the uterine wall, it is cut off from resources. right? It's like it doesn't have access to maternal resources yet because it's not yet in contact with the uterine wall to form a placenta. And so it's like cell division is happening, but the cells are just getting smaller and smaller and smaller. So the mass of the thing is not growing, is not going up. It's doing cell division, but each cell is getting smaller and smaller and smaller. Finally, after implantation and the beginning of innervation with the circulatory system of mom through the uterine wall, then the thing can actually start to gain mass, grow. The cells can start becoming bigger, to fuel more division. So, and this is sort of fits up with this because it's like, we're going to take one cell and turn it into two. And in order to get those two cells that are functioning and alive and working and all that other chemical stuff, once you shut the DNA down, you have to make sure everything is produced enough that you need to get started for the next go. The cell can still be smaller when the division happens, but you need all the resources you need in order to actually work. Right? So, and then if the cell actually works after division happens, it can just like make more of stuff. I'll make some more rough ER, I'll make some more Golgi, but in order to make more rough ER, you need some rough ER to get started with. So that's that. That's that. Giselle, did I answer your question or not?
That's it.
Okay. What was it? What was your question?
You said I was like,
What was your question anyway? Let's circle back to it. So this is all interphase right there. Mitotic phase, make this lovely baby blue color like this. And it consists of, I don't know what your textbook says, either 4 or 5 stages within. I go to 4, but some textbooks say 5. The first 2, just to explain how the difference might show up if you look through the textbook example, the first part is called, we'll get into details, the first part is called prophase, the second part is called metaphase. Some textbooks put this other one in there called pro-metaphase. I don't, it's the early middle phase. It's like what? It's like episode 1.5 of a sequel. It's like splitting hairs at that point. First one, and so this is moving all in this upward direction. Prophase. Characterized more than anything else by the breakdown,
That color isn't showing up.
the green,
it is.
Oh, yeah, it's just wild.
Yeah.
the breakdown of the nuclear envelope. So when we were looking at the nucleus, or when we were talking about the nucleus, and I was like, big thing, double membrane, super cool, packaged within this thing, right? That nuclear envelope starts to fall apart. It starts to fall apart. Eventually, you're going to take genetic material and you're going to divide it so it splits between 2 daughter cells. In order to do that, you need to get this nuclear envelope out of the way. Right, so it's like, let's break down the suitcase so we can actually get to the stuff inside. The next bit is referred to as, I really like this blue color. Sarah, do you like this blue color? It's up there. Do you see it? Okay, cool. What, are you watching the live stream? Okay, cool. You're not watching the live stream? Then what are you doing?
What is the title?
Who? Oh, I don't know. You have to follow me on YouTube to see it. That's PCF275 at email.bccs.edu. Metaphase. So what happens in metaphase? Paired, P-A-I-R-E-D, paired chromos line up in middle. And I will draw a picture of this. I will draw a picture of this here in a minute. I'm just doing words right now for context. Next, we have anaphase. Chromosomes divide to the poles. So they will actually start the separation process, moving from the center line of the cell out to the fringes. We have last one, telophase. Chromosomes start to unspool, unravel. They start to unpackage themselves so they can be accessed again. Slash nuclear envelope reforms. And then we're followed by one more thing that comes after that. Followed by cytokinesis, which is actual true cell division. That is when the one cell becomes two in the cytokinesis part. So when cytokinesis happens, you now have two cells. In what part of this context are those two cells?
Interface.
You now have two cells that are in
Interface.
interphase.
Going through the process.
We're at interphase.
Oh,
All this is interphase, right?
G1.
G1. So it's like now you have two cells which are right here, which is why it's like it's a line, but then it's also like a cycle. Yeah, it kind of becomes cyclical as well. This is the line version. Would you like to see the circle version? Let's see.
Can you
What did I?
Like the top part of the line, yes, it's cytokinesis. Yes.
Let me see it.
Gotcha, thank you.
What does it say at telophase?
It's where it splits.
Chromos
Unspool.
unspool. No Clegg envelope. Nuclear envelope.
Okay.
You got to be in it, man. You got to be in it, right? Many a joyous time has been spent with me in rooms where I don't have like big obnoxious tables in the front. I'll do the whole thing on chalk. Like in this front, it's like if you walked into the IMIC class and looked at the board, there's like I have no idea what the hell that is.
And
It's
what
like
is it?
you got to be there for it. I mean, you gotta watch the process unfold.
And for prophylaxis, does it break that a nuclear ew? Nuclear envelope.
Nuclear,
Ow.
nuclear,
Nuclear.
nuclear envelope. Welcome.
How did George Bush say it?
Nuclear,
Nuclear?
nuclear. This lecture is not brought to you by LaCroix.
Also, what is, I tried to find the channel.
I'll find it. We'll look it up here in a hot second. So,
If you go to like your module, the videos you've linked, you can just click on one of those and it'll take
yeah, it'll take you to all of them. Yeah, it'll take you to all of them. Let's see, what next? What next? Okay, circular version. Sound good? like this paper because it's thick, and I can do one on each side. Doing the hardest thing that you can do, draw a perfect circle on a piece of paper. Oof. I'm not going to do it well. You know why? Because I'm thinking about it. If I would have just done it without thinking about it, it would have been great. But once you start thinking about it, you already lost it.
Think about drawing this.
Think about my birthday. Think
Or
about
just go get a compass.
well, I tried thinking about my birthday earlier, and I didn't remember what I actually did. So, it's like,
Exactly.
so it works, Tavion, right? So it's like, yeah, that'll me thinking of my birthday will completely clear my mind of any higher thought, and I'll be able to draw what anyone would clearly describe as
You could draw a perfect.
a perfect circle.
You could draw a perfect circle by just tracing the LaCroix.
That's tracing, not drawing.
Yeah.
That's tracing. That's not bad.
That's pretty good.
That's not bad. That's pretty good. You just gotta,
Think about your birthday.
you just gotta go all in, man. You just gotta take the jump. Eat the elephant. Jump the shark with it.
You're gonna try that.
So that's your homework for next week. And, get a piece of paper out and try to draw perfect circles.
Isn't it illegal to eat an elephant?
In some places.
They're endangered.
Probably not that tasty either. All right. So if we kind of do the same thing on this, on such a circle like this one, we would say that this over here is, if you can draw a pretty decent circle, you're halfway to Charlie Brown, It's like just kind of draw a run circle, put a couple of hairs on the top of it. This one's a little more wider than it is tall. So from this side of that circle, it'll be interphase. On the other side, we will say this is mitotic phase. Okay. I'm going to draw another little hash like right there. Just for kicks. Most of the cells in your body are hanging out right here in G1. G1, pardon me. Cell's living its life. Best, most Insta-worthy, TikTokable life. And that's where it hangs out. That is, raise your hand if you have ever driven an automobile. Jessica, have you never driven a car?
What?
Have you never driven an automobile?
Oh.
Perfect. What is this, Jessica?
It's a stop sign.
What do you do in it? If you want to keep driving, you will stop right at the stop sign. There's a couple of places in the cell cycle. I did not put these on the linear one, right? But I am going to put them on this one. There's 3 places in the cell cycle where you have essentially what is literally referred to as stops. When you think about the cell cycle, a lot of times you think about it like you do the Krebs cycle, like you do Calvin-Benson, like you do a car or a wheel on something. It's like it's always kind of going around in circles and kind of moving and moving and moving. It is not. All right. Most of your cells are not always progressing through the cell cycle. Most of your cells are in G1 and they get to right about here and then they hit the brakes where it's going to hang out right here. And they do that for very good reasons. What comes after this is
G S.
S. What happens in S?
DNA divides.
DNA
Duplicates.
duplicates. That's okay. It's fine. Under what circumstances would DNA duplicate in a cell?
When it's getting ready to.
Yeah, the only time it's ever gonna do that is if it's gonna go through eventual mitosis and divide. If it's not gonna do that, because everything is fine, you don't need to make this DNA. You don't need to, start copying stuff. You don't need to, okay, we're gonna do all this copying of DNA, which is gonna be shockingly expensive. You don't need to do it. So it's like most cells are just like hanging out right here, sort of waiting for the light to turn green. If the right biochemical, body-wide environmental signals happen where it's like, okay, we need to make more copies of you, so divide, then this stop will go down and the cell will start progressing through. But most of it is like hanging out right here at this stop. Sound good?
Sound good.
Perfect. Remember these stops for later. They're going to get real important here in a bit. Super important. If you ask me, they will become super important. They may be less important to you, but they're important to me. And then we're going to start going through this next bit, which is S, right? Which is
Synthesis.
DNA synthesis, where we duplicate. We're going to duplicate the DNA. I'm going to say more statements about duplicating DNA here in a hot one. On page 3 of our drawings. Then we're going to move up into G2. This is cell gets ready to divide and mitote. I don't know if mitote's a word, but I use it, so I'm going to go with it. Mitote. And then whoa. Full stop. This stop is a, do we need to divide? Are we good? If we're good, we can stop here and we can hang out. We're just going to eat Fritos at the gas station off the Beltway and just kind of hang out at the rest stop kind of situation. Okay, we need to go. Perfect. Once you're done with G2 and go into mitotic phase over here, you better have everything done and made and constructed that you need in order to actually make it all the way through, right? So this is a stop, check the suitcases one more time, make sure you packed everything, because from here on out, it's a one-way trip. All right, so it's like,
Backing out because we're not stopping.
yeah, this is the check the truck now, make sure the bodies are out because we're not stopping once we go through this one. All right, so pause, do inventory, make sure you got everything you need, and we're ready to go. Mitosis. Can't use that color because it's similar to the other color that I used for the other thing that's the wrong color. Okay. So then we have, can you see this or is it too light?
We'll load it.
There's something going on.
Is it dark enough?
Yeah.
Prophase, metaphase, anaphase, telophase. I use a secret fancy, what do you call those? It's like when you have letters that try to remember, they have a name for each one,
Acronym.
acronym or whatever. Yeah, it's
It's
like, yeah, mnemonic, that's what it, yeah. Because the Linnaean hierarchy is like, I learned it as like kangaroos play checkers on fuzzy green squares. It's like K, P, C, O, F, G, S. And I've heard like other places in Fairfax County is like this, like King Philip does something. It's like, I don't know. Naomi's like, well,
Yeah. I don't know what it is.
I've heard them get kind of off color and filthy as well in terms of raunchy humor. So keep it clean, kids.
But the King,
Yeah, keep it
the
clean.
King Philip one was for like the animal orders.
It's all, yeah, the Kingdom, File, and Class Order,
Yeah.
Family, Kings, File, Linnaean hierarchy. Like kangaroos play checkers on fuzzy green squares. King Philip does something. I don't know what Naomi.
Why is it always?
Maybe,
King
maybe in
Philip?
the middle of the night, Naomi, 293, and you'll wake straight up in your bed and you'll go,
No.
"Ah," and you'll blurt it out once it comes back to you.
I don't think it'll ever.
I remember, for mitosis, I remember PMAT. And the way that I remember those is prophase, metaphase, anaphase, telophase. So it's like, that's my mnemonic for it. I don't know what to say about that, right? But it's like, it's only four. You can figure the PMAT, PMAT, I don't know. But you remember what these are, right? Prophase, this is where we, take the chromosomes that are all stranded out and we condense, right, chromosomes. Metaphase, we line them up. Anaphase, we split them. Telophase, NUC, separate. Let me say, I'm not going to separate nucleus. I'm going to say reform nucleus. You start building a nucleus again in telophase. And then this is going to be cytokinesis.
Can you say condense?
Yes. So we're like going this way, this way, this way. And when we have cytokinesis happen over here, we have the cell, great, which then goes over in a G1, but we also have another cell which comes over here and then starts its own G1, right? So it's like, if we follow this around, like circle goes around and around and around, we're following one of these two cells that come out of it. The other one kind of goes over here and starts its own cycle, right? So that's why it's like, that's why it's sort of part of the reason, like, I don't like the circular framework of this, because it could go this way, but it's like, if you see it as a cycle like this, you're always just like pairing one off to the side to sort of do its own thing. When in truth, what you're doing is this. You have the cell lineage that goes up and it divides, yes?
Yes.
And then they go up and then they divide. And then they go up and then they divide. Where you have, alternating. You have alternating, like interphase, mitotic phase. Then you have interphases, and then like mitotic phases. And then you have like interphases, and then you have like these right? So at each step along the way, you're alternating between interphase, mitotic phase, interphase, mitotic phase, interphase, mitotic phase. And each time you do that, you go from 1 to 2. So it's like, first I drew a line, and then I drew a circle. Now I'm drawing a branching diagram like this. Giselle, what's the truth here of all of this? One cell becomes many. One cell becomes many clones. So point to your liver. Right side under the ribcage. People are like doing low key. It's like, right here, right? Point to your left kidney, if you have one. Okay? So we're like this. Point to both at the same time. Yeah, can you do it?
I think so.
Sarah, are you pointing to both at the same time?
Since when were your kidneys in the back? I'm so confused.
They've always been in the back.
All right, fair enough.
They've always been back there.
All right.
We've been lying to you this whole time.
I thought it was like in your stomach.
It's not. It's not. They're about, kind of right there-ish, one on each side. Cool. So, every cell that is in my liver here, where's your liver hand? Right here. Every cell that's in your liver has all the genes that it needs in order to be a good kidney cell. All these cells in your kidneys have all the wonderful genes and DNA in order to be a very good liver cell. Why is that?
Because they sprouted from the same cell.
Sprouted?
They divided.
What's the right word here, though? Yeah, you're absolutely right. You're absolutely right. What's the right word? Duplicate, derived from, or
They might just.
mitoted from, descended from. I like that one. So what does this cell have to have? If this is like a human lineage, what does this cell have to have? One single cell. It needs to have
DNA.
everything.
Everything,
Every gene you need to do everything.
including DNA.
In especially the DNA. It needs the DNA to do everything, right? We call these cells, so it's like single cell, undifferentiated, has all the cells, all the genes in it that it needs in order to do everything. It has not yet started going down a pathway of development that will turn it into either a liver cell or a kidney cell. We have a fancy word for what those cells are. Anybody know what we call those? We call those
Stem cells.
stem cells. Stem cells. It's a cell that can be turned into anything. Because what it means, so it's like, let's say down here down the line, these cells are going to be liver cells. These are going to be kidney. These are going to be nervous tissue. These are going to be, I don't know, left arm cells beats me, right? What does this represent? Right? As you go through time, like time is obviously moving this way. This is apparently, this is a graph that demonstrates development, like going from a single cell to a fully formed multicellular thing, right? What happens at each one of these nodes? What happens there?
Less and less.
More stuff.
They all have the same DNA. What's happening as you go through time? They're dividing. Different genes are being flipped on and flipped off, just turned on and turned off, right? All of this, to go from a stem cell to a liver versus stem cell to a left arm versus stem cell to a nervous cell, it's what genes do I turn on in what place, at what time, in what order? Because the entirety of every cell in your body has them all. So it's like going from a stem cell to a liver is first I flip these on, then I turn this hit this bus line and turn all of these on in the circuit breaker, and then I wait 2 days, and then I flip this one on, that makes bile, and then I wait a couple of days, and I flip this one on, that puts it right here in the body, then I wait a little bit, and then I flip this one on, that puts blood through, So it's like, what order do you turn different genes on in the course of the history of dividing cells? And we call that development. That is human development. That is the development of anything, is what is the order of turning things on and off? What is the order of turning these genes on and off? But this is cool, because what this means is that with new technology, if we want another Tavion, or if we want another Jessica's awesome, we could use two. Right? The answer I'm looking for is, hell yeah. Hell yeah, she's kick-ass. It's like, we need two Jessicas. One is delightful, two would be more so. Not more so, what is good? Yeah, we're getting bogged down in the details here, Jessica. So how do we do that? So, once I know that every gene to make a Jessica is in every cell within Jessica, I just need to, Jessica, can I borrow some cells? You know, there's pluck one of the little hairs right off your head right there, you know. Snip it off, put it in a test tube, put it in a high calcium environment, apply a little electrical shock to it. All of a sudden that resets it back down to boom, stem cell stage. I can then put that in a nutrient-rich environment and it will start growing another Jessica. We can make clones of
Faster than
people.
you can say parthenogenesis, you have a.
Yeah, you have a brand new Jessica, right? So we can clone things. We can clone things this way, knowing this. It's like, well, if I can take this differentiated liver cell and genetically reset it so that it thinks it's all the way back here, then I can just implant it in somebody else's poor womb as a host, and I can grow and do all of it as it goes down the developmental pathway. There are some both ethical and biological considerations to make before making said clones. Just to be clear, right? Just to be clear.
So let's say you have one of these stem cells, you put it in a nutrient-rich environment.
Hypothetically speaking.
Hypothetically.
Hypothetically, you have one of these things. Asking for a friend.
And then in 9 months it would grow into a baby? Is that what you're saying? Or
Well, I mean, the environment, the environment sort of needs to be a little bit favorable to doing that, right? But this is, this is sort of the Jurassic Park thing, right? If you can get some dinosaur DNA out of a mosquito, you could grow it into a new dinosaur. That's not where the research is, because that's like, the DNA is just way too old and not really available. However, saber-toothed tigers, woolly mammoth, the auk, the extinct bird, the auk, a lot of extinct organisms where specimens are still preserved that you can get full genomes out of. There are scientists actively working on re-establishing the woolly mammoth as a thing. Is it a good idea? Earth's a little hotter now. It's not going to have any mammoth friends. Right? So all you do, I love it when it's like, all you do, like it's easy, find a female Asian elephant, take an egg from an ovary, ask first. You want to get consent on that. Find a cell of a woolly mammoth that has nuclear material intact, suck the nucleus out of it, take the egg, suck the half nucleus out that has half the DNA that it was thought it was going to pair with a male Asian elephant, take that whole nucleus from 20, 30,000 years ago, put it in the egg, calcium-rich solution, give it a little bit of a shock, off to the races, wait a week, implant it in Asian elephant mom's uterus, 22 months later, you got a woolly mammoth. I am glossing over some details here, but that's the gist of it. That's the gist of it. That's sort of the blueprint. What actually happens is not going to deviate far from that. All right. So, but the implications of this, that like every cell in your body has all the genes you need in order to be any other cell in the body, that's kind of cool. And all the research now is, if I can have a couple of your cells, I can grow a liver for you if you ever need one. It's like synthetic organ growth that is a match to your own genome because it's based on like your own cells, which is kind of neat. Or eyes or kidneys or sort of you sort of might need. And there are a couple of cool movies made about this. One was called The Island. Scarlett Johansson and the dude who plays Obi-Wan now. Ewan McGregor. Anybody see this one? Spoiler alert! Are you gonna watch it, or do you want me to spoil it?
Go ahead.
It's like you're not interested in that 30-year-old movies at this point. This is before Scarlett Johansson was like super famous, and there was this awesome community, this utopian futuristic community where all these people were blissfully happy and all their needs were cared for all the time, and there would be this lottery, and if you won the lottery, you got whisked off to this magical island. Where the rest of your life would be pure utopian bliss, and you were never seen again or heard from
I
again.
think I've seen this movie.
Yeah, and of course, what the lottery represents is what the individuals in this weird utopian community represent, or they are the clones of very rich people on Earth, right? And when they need a new like organ harvested, they would dial this place up, and their clone would win the lottery, and then have their organs harvested. to get transplanted into the rich bastard, right? Who made a clone to have them. Anyway,
I saw two similar movies to that.
here's a few, yeah.
Yeah, there was one with Andrew Garfield where they were like raised as children to be like organ donors.
Yeah,
And I think it was Carrie Williams. They were not-
yeah,
And
right.
there's a whole nother one.
There's a
Like,
few.
Similar concept. Too many.
Why not? Why not?
Similar
So,
to it, it's called Unwind.
mm,
It's basically it's kind of like it's a really really good book. It's kind of like on the lines of like abortion and like donating your organs and stuff like that. So like basically like you give up like abortions are out of the way at this point, and
right.
instead what you do is you have until your child is like 13. And like if you don't want your child all the way up to the age of 13, you can basically like just give your child away. And basically they are put into these concentration camps, and then they are deformed. So like they're still alive, but like their organs are off of them. And then like let's say like I was in that situation, right? And the Sim needed an arm, right? So like my arm is still mine, but it's attached to him.
Oh, interesting.
It's kind of weird, but
That
it's
is
a
weird,
really
yeah.
good book though.
Yeah, cool. Yeah, I'll check it out. I'll check it out. We're glossing over some ethics here, with all of this. The biggest one that I still have you for 2 more minutes. Hang on. The biggest one, of course, being does your clone have all the rights of a fully functioning, independent human, right? Is a clone made from your cells your child? Or are they not? Do you have, so it's like, where do you sort of start with that? And the way to start with that is, well, it's easier to say they are, or it's more difficult to say they are sort of your whatever, and then try to make a case that they're not. It's like, let's argue that your clone and any cells that come from you that result or end up being a fully formed separate entity are in no way your rights, responsibilities, property, anything whatsoever. Argue a case that they are, and you'd be like, So it's like a lot of people do it. It's like, well, of course it's mine, but then people say, well, these are reasons why it's not. I think the better argument is, of course they're not. Could you even begin to argue that they should be or that they could be? And this goes back to the what if you cloned Hitler conversation that anybody in any freshman college class or senior high school class always brings up. If we cloned Hitler, would we get another Hitler? No.
Technically, yes.
You would get another genetic Hitler, right? You would not get another behavioral, sociological, contextualized.
Yeah.
But how much do we know of Hitler that's not like nurture versus nature? That brings up that argument.
And also, if you are Hitler's clone, everyone knows you are.
Yeah, then wouldn't you?
Yeah, you gotta avoid-
I'm not saying you wouldn't go through the world with some baggage, right?
I don't think that should be like even like considered. Like why would we want to do that?
Want to clone?
Hitler or like Lenin, Stalin. Like why would we want to clone anybody? Like
You clone them so they
you
can
want to clone
try.
someone so like innocuous and like unseeming, like just some random.
We're all
person.
special on this world though right now.
But I'm saying someone that has
Not
a
yours,
mask.
Let's find the most banal, boring, average person on earth and clone them.
Someone who is not committed
And
like
that's
grand
sidesteps,
like full-scale crimes.
Good questions, though, good questions. Thanks, everybody. So I'll see you on Monday, correct? Awesome, and we'll talk a little about some of the details, a little bit this, a little bit more, and we'll talk about cancer.
Cancer.
Yeah. There were more people said yes to cancer than I would have thought.
Are we here or?
We are here.
So,